Hi gang! Today I'm doing the second part of the learning and memory lectures. So the last part ended with talking about, that's my parrot, you'll hear him from time to time, that's Rococo. The last the last lecture ended with us talking about HM, Henry Mollison, and I am going to post a little short video, not made by me, just to reinforce his story. But now I'm going to move on to talking more about how learning and memory is instantiated in the brain, like what's actually happening in the brain when you learn and remember things, what's happening on a neural level, what's happening on an anatomical level. So first I'm going to talk about different kinds of learning. I'm going to talk about associative learning first. And the two main kinds of associative learning that you've learned in your, Psych 100 classes and all your lower division classes includes classical conditioning, which is also called Pavlovian conditioning. And we're going to look at brain areas and what actually happens in the brain when you experience Pavlovian conditioning. And then we're also going to talk about operant conditioning, the other kind of associative learning that you learn about in your introductory psychology courses, and how those pathways differ from the classical conditioning pathways, and then we'll move into non-associative learning and see how those are different still. Okay, so first let me remind you what classical conditioning is. So you should already know this from your lower division work, and just generally speaking, if you have a psychology degree, if you have an undergraduate degree in psychology, you should know what classical conditioning is. It's all over, media and just in the general consciousness. So classical conditioning is also called Pavlovian conditioning because he's the one who first demonstrated it and wrote about it at length. And conditioning is just a fancy way of saying learning. If we are conditioning an animal, we're teaching it. The animal is learning. It's being conditioned to do some kind of new behavior. And in classical conditioning, we call it a form of associative learning because we are associating two things. We're associating one thing with another. And in the case of classical conditioning, we're associating two different kinds of stimuli. We're making stimulus-stimulus connections. And the first kind of stimulus that we need in order to do classical conditioning is what we call an unconditioned stimulus. And the unconditioned stimulus is any object or event, any kind of stimulus in the environment, any kind of thing or event in the environment, that when it occurs, humans or animals have a certain consistent, natural response to this object or event that is innate. It is, how would I say it, instinctual, or something that is programmed in our DNA for us to do. Something that you don't have to learn how to react to this stimulus. So for example, if something flies at your face, like somebody throws a rock at you or a ball at you or their fist at you, and it comes towards your face, the natural reaction that even a baby would have if something was coming towards his face, or any person who had normal reflexes, would dodge out of the way or at least blink and try to scrunch themselves up to brace for the impact. So those kinds of things, the thing flying at your face is an unconditioned stimulus. We have a natural response to it. And there are lots of things that are unconditioned stimuli in our world. Food that smells delicious when it's cooking, makes our mouth water. And we naturally crave those things. If something smells disgusting, usually the reason it smells disgusting is because it's an unconditioned stimulus, and we have an innate genetically pre-programmed reaction to those smells in order to get us to not want to eat or touch those things. So feces or something that's dead, these kinds of things can have lots of bacteria and germs that can harm us. And so it's good for us to be naturally disgusted by them so that we won't eat them or touch them. So there are all kinds of different things in our world that are unconditioned stimuli. And our natural response to unconditioned stimuli is called the unconditioned response. It's a response that you don't have to learn. So if something's flying at your face and you dodge out of the way, or you squinch up your eyes, you flinch, that's an unconditioned response. The flinching, the dodging, the squinching up your eyes. If you smell something disgusting and you want to move away, back away from it, that's an unconditioned response. That's a natural response. If you smell delicious food cooking, your mouth watering, that's an unconditioned response. You can't control that. So to classically condition someone, we need to make a stimulus-stimulus connection. We need to pair the unconditioned stimulus with what's previously a neutral stimulus. Well, what's a neutral stimulus? A neutral stimulus is any kind of stimulus that doesn't provoke a specific, consistent, innate response from people. So for example, music. There's all kinds of different music, and you can play any song and there will be a person who hates it, and there'll be a person who loves it, and there'll be a person in between who doesn't really care. So a song is an unconditioned stimulus. You have to learn whether you like a song or hate a song or what have you. This table sitting right here is an unconditioned stimulus. If you see a table in front of you, there's nothing naturally that you have to do with a table. There's no innate response. You see it, maybe you could set something on it, maybe you could knock it over. You could do a million different things with it. There's no specific innate thing that people do every time they see that item. And so we call that a neutral stimulus. And in classical conditioning, we are going to pair the neutral stimulus with the unconditioned stimulus. So we are going to take something like a song or a word, and we're going to pair it with, let's say, being punched in the face. Okay? So every time I punch you in the face, I say a certain word. I say, fluffy buttercup. Every time I punch you, I say fluffy buttercup. Okay? If every time I punch you, that's the unconditioned stimulus, I also say fluffy buttercup. That's the neutral stimulus. Eventually, you will flinch just when I say fluffy buttercup. I can take away the punch and just give you the neutral stimulus, and you will give me the same response as if the unconditioned stimulus was there. You will flinch to the sound of fluffy buttercup. That's called a conditioned response. You have now learned to have a response to something that was previously a neutral stimulus. And in that way, the neutral stimulus becomes the conditioned stimulus. And when you have that same response to the conditioned stimulus as if the unconditioned stimulus was there, then you have conditioning. You have a conditioned stimulus and a conditioned response. If that doesn't make sense to you, you should go over that and review it until it does. So you should review it in your book, or even if you have to go back to intro psych, or just play this again until you understand what I'm talking about, because it's about to get a little more complicated, because I'm going to tie that to the underlying neuroscience. Okay, so classical conditioning is something that occurs with humans all the time. In fact, phobias are one of the main kinds of classical conditioning that we run into in clinical psychology. So people have associated a fear response with an object or an animal that used to be a neutral stimulus. It doesn't necessarily provoke a consistent response on its own, but early on you were that object became associated with fear. So for example, fear of snakes, fear of spiders. What often happens is that early on a child sees an adult or an older child have a fear response to the snake or the spider or whatever it is, and then they adopt that same fear response. Because it is an unconditioned response that when we see other humans in distress, that we react in distress because it's helped us survive. If we see another human in distress, it probably means they're in danger or danger is around. So if we react to that, it helps us survive. So if another human is reacting to something, we naturally respond to that. And if that becomes associated with something that by itself would be innocent, sometimes people are afraid of, small animals like a hamster or a bird, all different kinds of things. Or even, if you watch Maury Povich, more unusual items. Like there was one person who was afraid of pickles and another person who was afraid of balloons. So you can have that classical conditioning response in any phobias. And they're actually really easy in the grand scheme of different things to help people deal with, because we understand how that connection works. It's also the same, interestingly, for fetishes, except for instead of connecting something fearful to the previously neutral stimulus, you connect something sexually arousing to the neutral stimulus. So for example, one of the most common fetishes is an underwear fetish, or a shoe fetish. And it's really easy to see because those two objects of clothing are frequently associated with, very sexualized, naked or nearly naked bodies. And, that kind of thing is naturally an unconditioned stimulus, the naked body, posed in a sexual way, unconditioned stimulus, and you associate it over and over again with something that by itself is just a piece of clothing, but you associate these two things again and again and again, and a person develops a fetish. So those are things that we commonly see, classical conditioning, forms of classical conditioning in psychology. So let me talk about those pathways in the brain, okay? Because, we can see that there are these different categories of behavior and these different kinds of learning. Well, does that mean that there are different kinds of connections being formed in the brain? And the answer is yes, there are different kinds of connections being formed in the brain. So let me describe what's going on in this, this diagram right here. What this is a depiction of is of a rat being classically conditioned to be afraid of a tone. Okay, so they want to play, usually it's just like a little beep, and they want to condition the rat to be afraid of the beep. So this is a form of classical conditioning. And what they do is they present an unconditioned stimulus. So the unconditioned stimulus is a shock to the foot. It's usually the bottom of the cage shocks their foot a mild amount, and that unconditioned stimulus, that shock, travels from their little foot through their thalamus, okay? And the thalamus, remember, is the relay station. The thalamus routes the information to other parts of the brain. So here we get a shock on the foot, it goes into the somatosensory thalamus, and then it gets passed on to two different places. One is the somatosensory cortex, so that you can feel the shock. But that information, being negative emotional information, ow, that hurt, also gets passed to the amygdala, and a connection forms to the amygdala. Now, at the same time, if I play a tone, that's going to be my conditioned stimulus. That's going to be what's previously a neutral stimulus. A tone is just neutral. If you play it for a mouse or a rat before it's been conditioned, you just play a tone, they won't do anything. They hear a beep, it doesn't make any difference. But when we're doing conditioning, we pair the beep with the shock. We do the beep at the same time we shock the foot. So the shock is traveling through the thalamus to the cortex, and the beep is traveling through the auditory thalamus to the auditory cortex. So the animal can say, oh, I heard that sound. It went to my auditory cortex, I consciously heard the sound. But the information also gets routed from the thalamus to the amygdala. And the amygdala is where we make an emotional connection, a connection about the pain, a connection about fear. And so that is how the connection is made between the conditioned stimulus and the unconditioned stimulus and the fear in the amygdala and the registry of I heard the sound and I felt the shock. So later on, after I pair those things together again and again and again and again, the rat becomes conditioned to learn that if I hear the tone, I'm going to get a shock. And so you can play just the tone alone, and you'll see the fear response from the rat, where it will either freeze or try to run to an area of the cage where it can't be shocked. Now here's an interesting thing about this. If you, after you do conditioning, let's say you do the whole conditioning process and now you have a rat that every time it hears a tone, it has a fear response, okay? It thinks it's going to be shocked, so it has a fear response. If you connect the projection to the amygdala, it will get rid of the fear response. Even though it learned to have a fear response, if the projection doesn't go to the amygdala, if it gets cut after the learning, the fear response goes away. However, if you leave that intact, if you leave the pathway from the thalamus to the amygdala intact, and you cut off the pathway to the auditory cortex, the rat or the mouse won't hear the sound. They won't be consciously aware of hearing the sound. It's as if they're deaf. But the information still goes to the amygdala and they'll still have a fear response. So after conditioning, even though the rat can't technically consciously hear the sound, it still has the response because the connection going to the amygdala is still present. Okay. Humans have a similar kind of paradigm that we can use if we're trying to do this kind of simple conditioning, this kind of classical conditioning in, it's simple motor conditioning. We're trying to get a response and it's just basic classical conditioning. With a person, we can't deliver shocks to the feet, generally speaking, due to ethical considerations. And so we have something that we can use that doesn't hurt the person at all, but it does develop a classical conditioning paradigm. In this one, what we do is we blast a puff of air to the eye, and at the same time as we blast the puff of air to the eye, we play a tone. Okay, so the puff of air to the eye, that information travels in through your somatosensory. It's somatosensory, it makes you blink. Okay, if somebody blows a little bit in your eye, just blows a little air in your eye like that, it makes you blink. That's it, doesn't hurt you, just makes you blink. If we blow a puff of air into your eye, it does the same kind of thing, it makes the same kind of connection as here, except for instead of this being the amygdala, it's the cerebellum. Okay, so for this simple motor conditioning that we're doing to people, there's no fear involved. We're not shocking them. Okay, so the place that this motor conditioning takes place is in the cerebellum in a place called the inner positus nucleus. And the inner positus nucleus is that spot in the cerebellum where this connection is going to form. So the unconditioned stimulus in this case is the puff of air. And it goes to the somatosensory thalamus and it gets routed to the somatosensory cortex and to the cerebellum. Okay? And again, the thing we want to be the conditioned stimulus is a tone. Eventually we want to be able to just play the tone and the person will blink if they just hear the tone and not necessarily have to have the puff of air in their eye. So the neutral stimulus, which is going to become the conditioned stimulus, is a tone, and that information travels through the auditory thalamus and to the auditory cortex and also gets connected to, in this case, the cerebellum, the inner positus nucleus. So that route, if you just replace cerebellum right here, is the route for that kind of classical conditioning paradigm that we use in people with the puff of air. Okay. Now I'm going to talk about operant conditioning. Okay, so operant conditioning is a little bit different than classical conditioning. Classical conditioning was about associating two stimuli together in order to get a certain response. We were associating a neutral stimulus with an unconditioned stimulus so that we could get the same response to the neutral stimulus as we get from the unconditioned stimulus, which eventually makes that stimulus the conditioned stimulus. Okay. That's pretty limited in terms of what we can do with behavior, because you have to always use an unconditioned stimulus. You have to have something there that people, or if you're working with animals always have a consistent, innate, inborn response to. And there's only so many objects that do that. With operant conditioning, what you do is you kind of wait for the animal to demonstrate a certain behavior, and then you give it consequences to its behavior. So this is about a behavior consequence association. And you can either reinforce the behavior, which is called a reward, making the behavior more likely to occur, or you can punish the behavior, where you make the behavior less likely to occur. Reinforcement strengthens the response, making it more likely. Punishment weakens the response, making it less likely. So if here comes let's say the little rat in the Skinner box, and there's a little lever there. If every time it pushes the lever, it gets reinforced by a food pellet coming out, it will keep pushing that food lever, that lever that dispenses food, every time it wants to, have a bite to eat. But if it pushes that lever and it shocks the bottom of its feet every time it pushes the lever, the rat will learn real quickly not to press that lever. Okay, so it's about consequences. And this is a classical paradigm for operant conditioning I shouldn't say classical. This is a well-known and well-established and well-used paradigm for operant conditioning in science. Okay, so where are these pathways for operant conditioning in the brain? Well, hopefully it won't surprise you that if what we're talking about is motor learning, that it's going to be the basal ganglia. That's very important, okay? We're learning new movements. And you'll remember that the pathway from the substantia nigra to the basal ganglia, that nigrostriatal pathway, is the one that's really important for learning new movements. And that the basal ganglia are the structures underneath our cortex, our subcortical structures right there. They're, limbic cortex. So right underneath the tippy top neocortex, we have these structures. The basal ganglia includes the caudate nucleus and the putamen. Those are called the striatum together. And they receive inputs from the cerebellum, specifically the caudate nucleus gets inputs from various association areas, because we're making associations. This is associative learning. We're associating behavior with reward. The putamen gets inputs from somatosensory association areas, somatosensory gyri. So we are integrating somatosensory information with this learning here in that's taking place using the striatum. So let me put this in terms of what's actually happening. Here's a tapir, and they're trying to train it to get its toenails filed, and they want to check it out at the zoo to make sure it's okay. So they try to get the tapir to go over in a certain direction, and when it goes over there, they give it a food reward. So now the tapir knows, hey, if I go over there when they're holding the little stick thing, they're going to give me treats that I like. And so then they file their nails and check them out and make sure they're okay while they're over there. So that's just a kind of simple operant conditioning. Hey, you came over here to get your nails trimmed, you get a reward. We do the same thing with our dogs. This is by far the easiest way to get a dog to do something that you want it to do. So dogs actually don't respond well to punishment at all. No animals really respond well to punishment. Usually when you use punishment, you get avoidance behaviors where the animal just knows it doesn't like that and so it runs away. But if you want to train it to do something, you have to give it a reward. So when the dog sits, when it finally sits, then you give it a reward. The same thing for potty training. Punishing a dog for already going to the bathroom on the floor does practically nothing, especially if any time has gone by. They don't have temporal contiguity, and they don't understand, why you're pointing at their poop and yelling. But if you take the animal outside and you take it for a walk and you wait until it goes to the bathroom and then you praise them and give them a treat and tell them, oh, they're such a good boy, then they know, oh, I did that outside. That's what I'm supposed to do. That's what I get praised for. And that's simple operant conditioning. And those associations are taking place in the basal ganglia of the dog. It's the signal booster to make those connections, so that those neurons now between that behavior and getting that reward or that consequence are strengthened in their brain. Okay, but there's other kinds of learning that do take place that's not associative learning. It's actually, there's not two things being associated with each other. It's not a behavior and a consequence. It's not a stimulus and another stimulus. It's just that something happens and it changes the animal's behavior in some way. So there aren't two things being associated. And the two that stand out the most and that we learned about the first are called habituation and sensitization. So the way that we figured out a habituation was a guy named Eric Kandel, and he's been in some of our videos. He's a little old white dude with big glasses and a big bow tie, and he talks like this. And he was actually in the video about HM saying, so when HM drew the star, he could see that he had drawn a beautiful star. Okay, that guy, Eric Kandel, I love him so much, I want him to be my Grampy. He won a Nobel Prize for this work because he was the first person who showed what's actually happening at a neural level, what's happening in the connection between two neurons when learning takes place. And the kind of learning he studied is called habituation. And so habituation is very simple. What they found was this: if you take a sea slug, okay, he had beautiful different kinds of sea slugs. I believe it was a plesia that he did this work with. If you take a sea slug and it's breathing underwater, okay, it has to open up its gills in order to breathe. But the gills are very sensitive, so if you poke them, they'll close, okay? So normally they're open when just the water's flowing around, and if they get poked, they'll close. Okay, so what he noticed is that if you took an aplysia and you sprayed a jet of water on its gills, at first the gills reacted as if they were being poked and they would curl up, they would close. But after a few seconds with the jet of water being on there, they would get used to, in some way, the water, and the gills would open back up. They would become habituated to the water and they would open back up. And Eric Kandel looked at that and he said, that slug doesn't even have a brain. That's a very simple animal. How is it getting used to the water? How does it know that it's okay for it to open its gills back up? And so he studied that mechanism. And it turns out that the mechanism is very simple in the slug. In the slug, what we have is we have a sensory neuron that picks up sensory information that's connected directly to a motor neuron that moves a muscle. There's nothing in between. There's no brain in between. This is connected to the gills collecting sensory information like, am I being touched? And this motor neuron is receiving information from that sensory neuron, and then if it goes to action potential, it's contracting this muscle. So there's no thinking about it, it's just sensory neuron connected to a motor neuron. And so what he found is that as the jet of water, when it was first applied, the sensory neuron would fire and the motor neuron would receive the neurotransmitter, and then it would get activated and it would fire and the muscle would contract. But after a while of the water spraying on the sensory neuron, a very small and important change occurred. What happened is that the sensory neuron would downregulate how much calcium could come into the end foot. It would make it harder for that calcium to come into the end foot. And if you make it harder for the calcium to come into the end foot, it makes it harder for the vesicles to bind, and that makes it harder to release neurotransmitter, and that means that that motor neuron isn't receiving as much, if any, neurotransmitter, and so it's not being affected. So the mechanism of habituation was simply turning down regulating the amount of calcium that came into the end foot of the presynaptic neuron. That's it. Turn down the calcium, turn down the neurotransmitter released, turns down the action potential in that postsynaptic cell. Now, sensitization is a little bit different and a little bit more unusual. Sensitization is where an organism actually becomes more likely to react after being presented with the stimulus. Okay, so you get a stimulus occurring, and after that stimulus occurs, the animal becomes more and more sensitive to it, meaning you need less and less of the stimulus to provoke the same response. And a really good example of it in human, because it's hard to think of that. Instead of getting used to a thing, you get more sensitive to it, but it does happen. And one example is in epilepsy. So having seizures through epilepsy. Early on in diagnosing a person with epilepsy, one of the first things they'll do is show you a strobe light, because strobe lights are known to trigger epileptic seizures. And so the first time a person who has epilepsy has a seizure, it usually takes quite a bit of flashing the strobe light before it will trigger a seizure. But subsequently after that, it becomes easier and easier to trigger a seizure with the strobe light. You can actually have, somebody could just see the flash from a picture camera if they've had epilepsy for a while, and it can trigger a seizure. So that can be a really serious situation. They can't have anybody around them even taking flash pictures for fear of triggering the seizure. And you shouldn't be surprised that what happens in sensitization is the exact opposite of what happens in habituation. In habituation, you down-regulate the amount of calcium that comes into the end foot. So in sensitization, you up-regulate the amount of calcium that comes into the end foot. So that's all that's happening, is too much calcium is coming into that end foot with an even smaller stimulus, and it makes an action potential much more likely, and in fact lots of action potentials, and that's what you have in a seizure. A more common, because epilepsy is, people have it, but it's fairly rare, but a really common kind of sensitization that occurs in drug use. So in drug use, you actually have an addiction, in addiction processes, you actually have both habituation and sensitization taking place, but in two totally different areas. The thing that you become habituated to, you develop tolerance to, is the substance itself. So the more you drink, the more you have to drink to get drunk. The more cocaine you take, the more you'll have to take later to get high. With opiates, it's a huge problem. You can develop tolerance within weeks, where you need more and more opiates in order to get pain relief, for example. So that's a huge problem with drug addiction. And so you can see, they get habituated to the drug, they need more and more of it to trigger the same feelings. But you get more and more sensitive to stimuli that trigger cravings for the drug. So for instance, if every time you go to buy drugs, you always have a big wad of cash, which is pretty common, if you see a big wad of cash, it might trigger your wanting of those drugs. Or if you have an alcohol abuse problem, you see those same people, you see your friends drinking at the bar, and you just crave it so badly you want to go sit with them and have a drink. So we become sensitized to those subtle cues surrounding the times and the circumstances in which we engaged in using those substances. And so you become even more sensitized to those triggers. And that's why they tell you in AA or NA that you should stay away from the areas where you used to use those substances and from the people that you used to use those substances with, because they don't want to trigger the sensitization process. Okay. So now I'm going to talk about some of the really classical ways that we previously looked at learning in the brain. So I've talked about, where we're making connections in the brain. I've talked a little bit about some neural mechanisms in non-associative learning. But now I want to talk about, first I'm going to talk about some early studies that gave us hints that we should be looking into the brain to see learning occur. And then specifically, one mechanism that's very well understood for learning and that you're going to need to know for this class. Okay, so let me talk about early on when we were first trying to figure out how does learning take place. Is learning, if we're teaching these rats to do something, are the muscles, do they somehow change? Did the muscles have memories? What is it about the animal that's changing that makes it able to learn? And so, by the 1800s, we're pretty well established that it's the brain that's changing, and we're doing a lot of experiments to see what is it that might be changing. And so here was an early experiment that got set up, and then the same paradigm was used over and over. The way that rats are normally housed in a laboratory is that you'll have a few rats all in one cage, 2, 3, 4 rats in one cage, and they get water and food at certain times of day. They get their bedding changed. They might have a little place where they can hide. They might get a couple of different kinds of food depending. And so you can create an environment for the rat, which we call an impoverished environment. And in that case, they would separate the rat out so that it was isolated. It would get no variation in food or bedding or lights. It just had a bare subsistence life where it got its food, its water, and that's it. Or we could put the rats in an enriched environment, which is, Club Med for rats, I guess, tons of different kinds of places where they can get their water, where they can hide and climb. They get toys, they get different kinds of food, maybe they get handled by the people who run the lab. There's lots of different ways that we can enrich the rats' lives. And so afterwards, after the rats lived their lives, they took their brains out and they weighed them. And that they found that the brain to body size ratio was highest in the rats that lived in the enriched environment and lowest in the rats that lived in the impoverished environment, and somewhere in between for the ones that lived in the normal environment. Then we figured out how to measure not just the whole brain, but the volume of the cortex itself. So they took out the volume of the cortex itself, that thinky, foldy part of the brain, and compared volume of cortex, to overall volume of brain. And they found out, again, rats in the enriched environment had more cortex to brain ratio than the rats in the normal condition, who had somewhere in the middle, and the rats in the impoverished condition had the smallest cortex to brain ratio. And then they've done things even with neurotransmitters and neurotransmitter indicators. So for example, we assume that the more acetylcholine esterase, ACHA-E, that you have in the brain means the more acetylcholine you have in the brain, okay? So they figured out a way to measure the amount of acetylcholine esterase in the brain, and they found out who has the most, the guys in the enriched environment. Who has the least, the guys in the impoverished environment. And who's in the middle, the guys in the normal environment. So even when it comes to the neurotransmitters themselves and the things that work with the neurotransmitter, the whole neurotransmitter systems are being increased by giving the rats these enriched environments. So what is it that is actually happening when these cells are going through learning? Okay, so the mechanism that most of your cells go through and that we have well established as the mechanism for most learning, the mechanism for plasticity in the human brain, is called LTP, or long-term potentiation. And LTP is something that takes place all over the brain, but we have studied it extensively in the hippocampus because, as we talked about last time, the hippocampus is that crucial area for consolidating explicit memories. Okay, so the primary neurotransmitter that is at these synapses is glutamate. So here's 2 cells. This is supposed to be 2 cells, let's say in the hippocampus, okay? And they are undergoing learning, okay? So let's say it's classical conditioning, something as simple as classical conditioning. So let's say that, a dog is learning to salivate to the sound of the bell, right? That's what Pavlov said. Okay, so in the dog's hippocampus and in his cortex, when the dog hears the sound of the bell, the presynaptic neuron might release some glutamate, and the glutamate might attach to the postsynaptic receptors, the receptors on the postsynaptic cell. Also, if the dog sees that its food is coming, okay, here comes its food, some glutamate might be released, and the glutamate might bind to the receptors on the postsynaptic cell, okay? So a stimulus might cause some amount of glutamate to release. In an excitatory postsynaptic potential, okay, glutamate is the main excitatory neurotransmitter in our brain. So the presynaptic cell releases the glutamate, it binds the receptors on the postsynaptic cell. That's an excitatory postsynaptic potential. We're talking about glutamate, an excitatory neurotransmitter. It lets in what ion? Sodium, the main excitatory neurotransmitter. And the kinds of receptors it binds to, we've talked about in this class before very briefly, are called AMPA receptors. So at these junctions, there's a receptor called an AMPA, and the R stands for receptor, AMPA receptor. And AMPA receptors are receptive to glutamate. When glutamate binds to an AMPA receptor, it opens up an ion channel for sodium, and sodium comes in. So that's what happens every time your dog gets fed food, okay? The food is very exciting, the glutamate gets released, it binds to the AMPA receptor opens up its ion gate, and sodium comes in, and it excites the cell, and the dog starts drooling or whatever. But there's another kind of receptor in that same synapse that glutamate can also bind to, but it's blocked by a magnesium ion. These are called NMDA receptors, and they are crucial for learning. It is crucial that you have these NMDA receptors and that they are blocked by a magnesium ion, because it's the change that's going to make learning occur. So you have a first stimulus, like let's say it's the sound of a bell, and the dog doesn't do anything, it just hears the sound of the bell. But now you put in a second stimulus, a bowl of food, and even more glutamate gets released by the presynaptic cell. Now you have the presynaptic cell releasing all kinds of glutamate. It's got a first stimulus, the bell, it's got a second stimulus, the food, and now you're definitely going to action potential, more glutamate has been released. But it's not just that the secondary cell goes to action potential. Another thing happens. What happens is so much sodium builds up inside the postsynaptic cell that it repels the positive magnesium ion out of its place. It pops the positive magnesium ion out of place because it gets so positive in here, it pops that magnesium receptor out. And now glutamate has 2 places to bind. It can bind on the AMPA receptor and let sodium in, but it can also bind on the NMDA receptors. And the NMDA receptors do something very important. They have a metabotropic potential. they can send a second messenger to the nucleus of the cell. And that second messenger tells the nucleus of the cell, build more AMPA receptors. We're using glutamate here a lot. We're using this connection a lot. You should build more AMPA receptors so that we can receive more glutamate. And so that's exactly what your brain does. It builds more AMPA receptors and it inserts them here in the end feet. And then the next time glutamate is released, it has a whole bunch more AMPA receptors to bind to. And that makes it way easier to send this postsynaptic cell to action potential. And so now you can play just the tone and the dog will react by salivating to just the tone. Because you've put so many AMPA receptors in here, all it takes is that one little stimulus and boom, your secondary cell, your postsynaptic cell goes to action potential. And now I have a little video that says that the exact same thing. It's a little bit different way of saying it, and it has animations, so I'm gonna click on over to that and let you see that. There it is.
The human brain is capable of storing information for as little as 30 seconds in short-term memory, or a lifetime in long-term memory. Think about the last time you studied for a test. The process of studying activates various pathways within your brain. The continued activation of these pathways is what leads to memory retention for long periods of time. Long-term memory has been associated with a horseshoe-shaped structure within the brain referred to as the hippocampus. This structure is found in both hemispheres of the brain within the medial temporal lobe and coordinates the storage and retrieval of memories. A closer look at the hippocampus reveals some major structures: the dentate gyrus, the CA1 and CA3 regions. Memory processing begins when signals enter the dentate gyrus from the entorhinal cortex via axons of the perforant pathway. Granule neurons, a type of cell within the dentate gyrus, synapse onto pyramidal neurons within the CA3 region. The CA3 neurons connect their axons, referred to as Schaefer collaterals, to the CA1 pyramidal cells. From here, the information is passed to the subiculum and then back out to the entorhinal cortex. A process referred to as long-term potentiation, LTP, is thought to be the cellular and molecular underpinnings of memory. LTP is a long-lasting enhancement in signal transmission between two neurons after repeated stimulation. LTP has been most commonly studied at the synapse between the Schaefer collateral axons of the CA3 neurons and the CA1 pyramidal cells. A closer look at the CA3-CA1 synapse reveals various structures that are involved in LTP. On the postsynaptic cell are both NMDA and AMPA receptors. These receptors are usually localized together at many postsynaptic sites and are activated after the binding of the neurotransmitter glutamate. The AMPA receptor is permeable to sodium ions. The NMDA receptor is also permeable to sodium, but more importantly, it has a high permeability for calcium. The NMDA receptor is also blocked by magnesium, which prevents the ions from passing through the receptor. When an action potential travels down the Schaefer collaterals, it leads to the release of glutamate, which binds to both the NMDA and AMPA receptors. When a low-frequency action potential is propagated down the Schaefer collaterals, a small amount of glutamate is released. The AMPA receptors will open and allow an influx of sodium into the CA1 postsynaptic cell. This causes a slight depolarization event in the postsynaptic cell. The glutamate also binds to the NMDA receptors, but no ions will pass through the pore due to the magnesium blockade. While the small amount of neurotransmitter release signals a response, it is not enough to cause LTP. This is comparable to you studying material for only a few times. You will not activate a high-frequency action potential from studying for just a short while. When a high-frequency action potential travels down the Schaefer collaterals to the terminal, a larger amount of glutamate is released from the presynaptic terminals. This is comparable to you studying for longer periods. This results in higher frequency action potentials. When glutamate binds to the AMPA receptor, a greater depolarization event occurs. The AMPA receptors remain open longer due to the increased concentration of glutamate, which allows a larger amount of sodium to enter through the AMPA receptors. This influx of sodium causes a large depolarization event in the postsynaptic cells, which repels the magnesium blockade from the NMDA receptors through a process known as electrostatic repulsion. At this point, the NMDA receptor, with glutamate bound, allows sodium and calcium to enter through its pore. In this way, NMDA receptors are known as coincidence detectors because they require a presynaptic and postsynaptic event for channel opening. The binding of presynaptically released glutamate and a significant postsynaptic depolarization via activation of the AMPA receptors. As previously mentioned, LTP is the process of strengthening the connections between two neurons. The influx of postsynaptic calcium acts as an important secondary messenger, activating many secondary intracellular cascades. The increase in calcium contributes to two phases of LTP, early phase and late phase. During the early phase, the calcium binds to its respective binding proteins and causes the insertion of new AMPA receptors onto the postsynaptic cell membrane at the active CA3-CA1 synapse. These AMPA receptors are stored in the postsynaptic CA1 internal cell stores and will only insert when there is a large influx of calcium through the NMDA receptor. This allows more AMPA receptors to be available for future depolarization events. The early phase changes last for only a few hours and require a brief increase in calcium levels. During the late phase, a prolonged influx of calcium causes an increase in transcription factors, ultimately resulting in gene expression and new proteins to be synthesized. Some proteins resulting from this process include AMPA receptors, which are inserted into the postsynaptic cell membrane at the synapse. In addition, there is an increase in the synthesis of proteins called growth factors. These are involved in the formation of new synapses, which is the basis for synaptic plasticity. Your brain can change as you learn. These synapses are formed between the CA3-CA1 neurons, allowing for a stronger connection between the two neurons. The late phase changes can last 24 hours up to a lifetime. It is important to remember that LTP is not a mechanism. It is an outcome of the increased activity between two neurons. This results in an increase in AMPA receptors and synaptic connections, which allows the low-frequency action potential to cause a greater depolarization event in the postsynaptic terminals. This is the foundation of memory. However, the hippocampus is not the only brain region that processes memories. Memory processing occurs in many other regions, including parts of the cerebral cortex. Studying activates various pathways throughout your entire brain. Continuous activation of the same pathways will create high-frequency presynaptic action potentials and postsynaptic activation in those paths. It is now clear that these pre- and postsynaptic events strengthen the connections in the specific pathway. This strengthening is what contributes to your ability to recall the material when writing your exam.
So I really like that video because it talks about what's happening in your brain when you study. So what's happening in your brain when you study for this class is that the more you study, the more glutamate you release, the more AMPA activation you're triggering while you're doing the studying. And what you're hoping is that you're doing this so much and so much and so much that you're causing enough of a depolarization event that the NMDA receptor is also activated, and you want it to make more AMPA receptors, put more AMPA receptors in, which is going to make it easier for you to then remember that information for the test. And then if you study quite a lot and it gets consolidated by good sleep at night, your DNA is actually going to be instructed to build even more AMPA receptors, which are going to get inserted there. And now you have a very strong neural connection. And the more you do this, the more you strengthen it and the faster it becomes available for you. And this is true for everything you ever learn. This is how people get good at things. They are building more AMPA receptors and making those neurons fire much more easily and in a much more fluid way. And then, like the video said, if you do enough of this, you can even stimulate the nucleus, the DNA, to produce growth factors and build whole new synapses. And so now you can form connections, scaffolding with other cells, which makes it even easier to grasp that information. So the reason that your professors can bring information to mind so readily, especially the ones who've been doing it for a while and practicing the same things, is because we've just strengthened these connections over and over again. We have tons of AMPA receptors, and in fact you can get so many, and you can, another thing that can happen is that the synapse can get closer and closer and closer together. So the synapse gets smaller and smaller. The presynaptic cell gets closer and closer to the postsynaptic cell. And eventually the presynaptic cell is releasing so much glutamate, and the postsynaptic cell has so many AMPA receptors for it that they call it a perforated synapse, because it's basically just like the glutamate is constantly releasing. It's super duper easy. It's like there's a perforation. It's like there's a hole that the glutamate's leaking through. And that's, what happens in things that are extremely well rehearsed in your brain. Like, the synapse for knowing your name. Or any other thing that you do just constantly. If you're the kind of person who does hacky sack all day and you're really, really good at hacky sack, super good, or ping pong, or whatever your thing is, basketball. The reason Serena Williams is as good as she is at tennis is because she has practiced and practiced and practiced so many times that she's built up all of these AMPA receptors and glutamate system in her brain, and so now she's expert at it. It comes easily to her. It comes automatically to her because she's practiced it. And that's true for anything you practice. Anything you practice is going to happen that way. So this is just showing you that same thing again, showing you the AMPA receptors and the NMDA receptors and the different kinds that you can have. And here you see the calcium coming in through the NMDA receptor, but not the AMPA receptor. And so that's the one where you can, have that calcium kinase trigger those second messenger systems, including inserting already built AMPA receptors and activating the genes to build more AMPA receptors. Another thing that can actually happen is that NMDA receptors can increase, so NMDA receptors can increase nitric oxide activity in the postsynaptic cell. And nitric oxide is a gas. So when it triggers the nitric oxide to be released, it can actually cross right through the cell membrane, right back across the synapse, and it can cause the presynaptic cell to release even more glutamate. So that's another way that you can have learning. The last thing I wanted to talk about a little bit is dendritic growth and then constraints on learning. So dendritic growth is exactly what it sounds like. It's when whole new dendrites are created. A new dendrite will come out and a new end foot will come out to connect with it, and it will create actual new connections between the cells that weren't there before. That's how you learn new things. And we've seen dendritic growth in animals. So this is a rat, and they showed a single cell with all of its dendrites sticking off of it, and how after training, okay, so they trained it to run mazes. They trained the rats to run mazes. They would grow new dendrites. You could see new filaments starting to come out in different places. And so that, confirmed for us, yes, indeed, whole new synapses can be formed. Whole new dendrites can grow. Another kind of aspect that's important to learning is pruning inappropriate connections. So, for example, the baby's brain is full of all kinds of different connections so that that baby can adapt its brain to whatever life it's born into. And so the connections that are not appropriate for that baby in its life and what it learns, those connections are pruned away. Those are inappropriate connections, and so they're pruned away. And we see that with things like language learning. And it's important so that it can maximize your ability to use your own language, use your native language. Another thing that can happen is that the structure of glial cells can change, and that can be a form of learning. So for example, you can have glial cells that, like I say, make the synapse smaller by bringing together the postsynaptic cell and the presynaptic cell. They can make that synapse tighter, and so glial cells can have an effect on learning too. But the very last thing I want to mention is that there are constraints on learning, not just because of an animal's body, but because its brain is constructed to control that specific body. A human being can never, without some kind of plane or something like that, a human being on its own cannot learn to fly. No matter how much a bird tries to teach you to flap your arms, it's not going to work. No matter they'll tell you to use this muscle or that muscle, it's never going to work because we don't have the physical structure to be able to fly. A cat doesn't have the brain structure to learn to juggle. It's just not part of a thing you can ever teach a cat to do. Its connection to its arms, from its brain to its arms, and its motor activity is not designed to do that set of skills, and so you're just going to frustrate yourself. An alligator or a crocodile is never going to be able to ponder justice and, philosophical matters of, what's right and wrong, because they don't have a cortex. Limbic system is the end of it. No neocortex. You're not going to be out there thinking about, how to have empathy and things like that. It's just not part of their bag. Dolphins live in a 3D environment. So human beings, we're just generally on one plane, but dolphins can move up and down and back and forth and every which way in the water. So they're used to a three-dimensional world, which actually means that they can't learn the difference between left and right, because left and right doesn't make any sense in that three-dimensional world, the concept of left versus right. And so animal trainers were really baffled for a long time why they couldn't teach that to dolphins, because they seem so very brilliant and capable of doing things. But then they realized, their brains just aren't set up to get that concept the way ours are, because ours are actually more limited than theirs. And so they would teach a dolphin something else, like that side of the pool or that side of the pool, and they could definitely understand those things. So, the same can be said for humans. There are constraints on our learning in our brains that we probably don't even know are there. And that, maybe some, super complicated alien intelligent life that we meet someday is able to, have connections that we can't imagine. I like to think about those things. Not that we have any evidence for that, but it's just fun to think about. Okay, so I'm going to post this now, and that's the end of the memory and learning. Thank you, and take care.